Hi students, welcome to the fourth video module of the molecular basis of inheritance. So in the previous three modules we have seen the replication, transcription and the structure of DNA. Okay. Now we will be discussing about the genetic code. Okay. This is a very important topic in NEET. Okay. So basically what is the genetic code? The process of translation requires a transfer of gene information from a polymer of nucleotides to synthesize a polymer of amino acids. As I said, it is called translation. Right, so there were some combination of base pairs that encoded the proteins, right? So these are called the genetic code. It was the George Gamow, a physicist, who argued that there are only four bases, that is A, U, G, C, right? And if they have to code for 20 amino acids, the code should be constitute a combination of bases, right? And he suggested that in order to form the code, the code should be made up of 3 nucleotides. So a total 4 cube number of propositions, 4 cube, right? You have done these things in the mathematics, permutation combinations. Okay? So 4 cube is equal to 64, right? So 64 total codons are there, but only 20 amino acids are there. So there are more excessive codons for a amino acid. Okay. The codon was a triplet. It was a proof, right? But the chemical method of development was done by Hargobind Khurana, a very famous Indian scientist. You need to remember the name. The chemical method was developed by Hargobind Khurana. Okay. You can see the table here. You can see that UUU codes for phenylalanine, UUC is phenylalanine, UUA is leucine. So basically you can see कि like पहला दो जो भी हो, right? UUA हो सकता है, UUG हो सकता है, right? UUC हो सकता है. तो इसमें क्या होता है कि लेट्स से तुम अगर लास्ट का थोड़ा चेंज कर दो कोडॉन, तो ओवरऑल अमाइनो एसिड चेंज नहीं होता. सो द लास्ट बेस डजंट चेंज द अमाइनो एसिड. ओके, so this is known as Wobble's hypothesis. So what are the salient features of a genetic code? The codes are triplet, right? And 3 codons do not code for any amino acids. They are called stop codons. They are basically UAA, UAG, UGA. These are called the stop codons, right? Some amino acids are coded more than one codon, hence the code is degenerate, right? So the code is degenerate. You need to remember this term, degenerate. The code is universal. Code have no punctuations. AUG has dual function. It codes for methionine, right? It's code of methionine, and it is also acts as an initiator codon. So the initiation codon is AUG and the stop codon is UAA, UAG, UGA. Okay. See, which of the following features of genetic codes allow bacteria to produce human insulin by recombinant DNA? Okay, genetic code is specific. This is correct. Genetic code is not ambiguous. This is also correct. Genetic code is nearly universal. That is also correct. So what they have said कि which of the following features the genetic code allows bacteria to produce human insulin. तो bacteria जब human insulin बनाएगा, so that means क्या कि the code is universal, right? Code अगर universal नहीं होगा तो हम कैसे बनाएंगे proteins from a different organism? तो आंसर होगा ये वाला. Genetic code is nearly universal. Okay. Which of the following is not a feature of a genetic code? Triplet, yes. Degenerate, yes. Non-overlapping, yes. Ambiguous, nahi. It is not ambiguous. It is unambiguous. Okay. So your answer will be D. Okay. The codon AUG codes for, as I said, AUG is a start codon, right? Or initiation codon. So it will be a methionine. Here comes the mutations. Okay. So we have seen a classical example of mutation in our principles of inheritance wala video. That is your sickle cell anemia, where GUG transforms into GAG. Insertion or deletion of one or more bases changes from the reading frame to the point of the insertion or deletion. So this is known as frame shift mutation. Can you see here? Ram has red cap, right? When you enter something like B, so what does it spell? Ram has B, DCA, P. Same as same as this, and then you enter this, then it says Ram has big red cap. So the frame of reading changes with insertion and deletion of any base pair. So this is called the frame shift mutation. Finally, the structure of tRNA. Basically, tRNA is a cloverleaf model. It has 4 arms. Okay, this is the codon arm which codes for the amino acids, and the opposite to the codon arm, that is called anticodon loop. Right, this is called anticodon loop, which is complementary to the code. Okay, and there are amino acids acceptor end, and which it binds to the amino acids and tRNA specific. Okay, and it is the initiated tRNA. Okay, so basically it was all about your genetic codes and all. Okay, in the next video module we shall be discussing about the translation, and in the next video module we shall be discussing lac operon and the human genome project. Okay, thank you.